Hello, everybody. We'll be online at 2 p.m. sharp. So just before I introduce to you our next speaker, I just want to thank you our sponsors, so Tor Labs and Lesco in Bonter, that they have give the opportunity to us to share, to have food and coffee. So all the coffee breaks are sponsored by Tor Labs, Alesco, and in Bonter. And also, of course, we need to say thank you to our funding agencies, so FAPESP and FAEPA, who help us to build this workshop. So our next speaker, it's my great pleasure to introduce to you Professor Alex Surjet. I don't know if I'm pronouncing Surjet. He is an associate professor of neuroscience at the University of Tours, France, and a member of UMR 123 iBrain. His research focuses on uncovering the neurobiological mechanism that governs stress integration, stress-related disorder, and therapeutic response, with a particular emphasis on hippocampal function. He made significant contributions to the field of neuroscience by demonstrating the functional role of hippocampal neurogenesis in the actions of antidepressants and in the behavioral and physiological response to stress. His current research aims to develop a mechanism framework for understanding population dynamics within the hippocampal formation and how these circuitries interact with a broader brain system to generate both normal and pathological behaviors. Professor Surjet's works particularly address how stress alters hippocampal and interconnected networks, contributing to the conditions such as anxiety disorders, PTSD, and major depressive disorders. Alex, thank you so much for coming all the way from Tours to Ribeirão Preto, and thank you for being so kind, sharing your research, and to be here with us this afternoon. Thank you.
So thank you for the nice introduction, and so I want to thank you also for the invitation and the opportunity to present here, to be here with you, and to present one line of my research, the longest one, right? Because this one with the UCMS that I will present, and neurogenesis stuff, I started with my PhD. So it's the longest line of research that I developed in my career. So, with the workshop topic, so I focus on the unpredictable chronic mild stress, explaining why I start using it, particularly in the field of adult hippocampal neurogenesis, and then to see how this procedure can also have the variability we can observe with this procedure and what we observe in the lab over the years. So first, I will try to change. Okay. So first, why I did start using chronic stress. So I will present that first, I told you, and then I will show the scientific result we obtained with this model, particularly the part on hippocampal neurogenesis, and then we will discuss all the variability. So first, sorry, I tried to change. Right. Okay. It works. Okay, so why I did start? So as I told you, I started during even my master's degree and the PhD. We started at this moment in the early 2000 to know that adult hippocampal neurogenesis can happen. So we have this neural stem cell in two restricted brain area, and particularly one is. I try if we see something. So this one, which is the subgranular zone of the hippocampus, where you have neural stem cells that can proliferate first and have proliferation, and then can produce newborn neurons that will integrate the dentate gyrus. And we have also, particularly in rodents, the subventricular zone that also can produce newborn neurons in the olfactive bulb after remigration. So we have this neural stem cells still active in just this restricted area. And I was interested at this moment what will be the functional role of adult hippocampal neurogenesis. So we start, first to correlate the change of the neurogenesis level, but we wanted directly to manipulate this process to see the impact on behavior and on functions that was important for hippocampus. So we can use at this moment pharmacological inhibition with MAM, for example, compounds that can inhibit the neurogenesis. We can use X-ray irradiations, focal on the hippocampus. It was I used the most in my studies. And there is also, after some years also, some transgenic animals where you can also induce an ablation of, an inhibition of neural, adult hippocampal neurogenesis. So some groups and our group start to understand. Hippocampus is also very known for his role in spatial navigation, memory, spatial and episodic memory. So many people try to inhibit neurogenesis at this moment and to see the impact on memory or spatial memory, spatial navigation. So you have a lot, and many find some result, but each group find a very specific aspect of hippocampal task, hippocampal dependent task related to memory, and that was not always the same. So some presentation here, some example of paper that was published at this moment. Some find it some very specific discrimination between two contexts. Some was like contextual fear conditioning, but those cannot find it. So actually it was a little a mess of the result. And some years after, voilà, there is even a meta-analysis, and you have here the result of the meta-analysis. Bon. I cannot move this. Ah, okay, it's good. So voilà, you have here the red line is when you have no effect, no combined effect size of the effect, and you can see that actually when you combine all the effect sizes here, there is no, on contextual fear conditioning or Maurice Watermace, for example, over all the years there is no significant impact on this function in hippocampal function. So actually, donc, inhibition of neurogenesis does not fundamentally disrupt. I'm fighting with this, huh? So voilà. Does not disrupt the hippocampus dependent learning and memory as well as spatial processing. We just have some specific aspect, like marginal aspect in hippocampal dependent function in this aspect. So then also people and we try to see when we inhibit neurogenesis, we can impact directly more anxiety, novelty exploration, and emotional reactivity. And some group tried, so some result was positive, like the group of Nora Bruce find something in elevated pulse maze by inhibiting neurogenesis with a transgenic model, but some other not in some specific tasks, not like NSF and like from swim test, others find other things. It was a little the same picture in this aspect on this kind of behavior. So, and again, there is a meta-analysis for growth groups. And again, when you look anxiety tests, you did not find over the year a combined effect size that is significant, different from no effect. So again, no functional involvement of neurogenesis in some hippocampal main dependent function. So if we see inhibition of neurogenesis only marginally disrupt main hippocampal function. We have some aspect in pattern separation. I will not go to this aspect today, but no direct role in supporting place cell activity, representation of safe location, which is very important for hippocampal function. No effect, like significant effect in learning and memory, which is like a very classic hippocampal dependent function, and also no effect on baseline emotional reactivity and anxiety, or only marginally in some group, in some model. So the question remains at this moment whether this hippocampus neurogenesis is just a vestigial remnant of evolution, like an epiphenomena of evolution that stays on neural stem cell is active, but with no real functionally relevant function. Or there is some function that we cannot see. So at the same moment, we also know that, when I started actually, that stress and glucocorticoids can decrease also neurogenesis in hippocampus. And on the contrary, learning, exercise, and also antidepressant treatment can increase it. So it was just correlated, but we wanted also to test this aspect that at this moment was not really related to hippocampus. Hippocampus was not seen at this moment as something relevant for depression and antidepressant. but there is this correlation. So starting to arise what they call at this moment the neurogenic hypothesis of depression, where maybe a decrease of neurogenesis causes the etiopathogenesis, or at least a vulnerability for depression, and the increase of neurogenesis could underlie some therapeutic effect of antidepressant. So so we wanted to test that, and to really, as objectives, to test the link between neurogenesis and etiopathogenesis, first aspect, link between neurogenesis and the behavioral effect of antidepressant, and then, if there is something, to see the functional involvement in neurophysiological processes associated to this behavioral effect. But for this, we need a model of depression with high validity, because we see that in baseline, when you remove neurogenesis, we have no effect. And we have some with antidepressants that we find, but still. So we try to to send some effect, and we wanted to find a model that maybe can fulfill this validity criteria from Paul Wilner at this moment, to have the construct validity, high construct validity, which represents the homology with the disorder, usually the degree of similarity between the etiology and the mechanisms that underlie the model and the disorder. The phase validity, which is the isomorphisms of the disorder, so which means the degree of phenomenological similarity between the model and the disorder, so the symptoms. If the model can reproduce the symptoms, or all the spectrum of symptoms we can have with this disorder, and predictive validity, which is the ability to predict the efficacy and the efficiency of the treatment. There is better model, but this is more simple for the sake of the talk, but there's more specific validity model, but So at this moment, we were on this. And there is many models of depression at this moment, also the LPS induction, bulbectomy, you know maybe all that, and maybe the other presentation will go into other models. So we were looking for the UCMS, which is the one that attracted our attention at this moment, and I was, and I'm still, with Catherine Belzung, which was my supervisor, that was starting also in the lab. We started to develop chronic stress with UCMS in mouse, because at this moment it was more known for rats and much less used in mouse, so we wanted also, in our model was in mice, to test it, so we start to develop that, the UCMS model. So the UCMS means the unpredictable chronic mice stress. And we wanted first, is good construct validity? Theoretically, yes, because it mimics the role of socio-environmental stressor to precipitate the disorder. And actually, chronic stress is a very important risk factor for depression. So in that aspect, we have an etiological factor that in the model that is also important for the disorder. So we have, for example, for the mouse, it was really socio-environmental, so we alter the bedding, sawdust change, sawdust removal, damp sawdust, we put water, we also use like cat or rat feces in some procedure, alter environment, catch tilting, it's a lot, shaking, light dark changes, predator sound, and social stressor. So we have a lot, and it can change from a lab to another, and even, you will see at the end of the talk, even in our lab, with the time, it changes sometimes, which is also a problem for viability and reproducibility, but it changed a lot. Also, regarding the mechanism, at this time we have access to a brain bank of human. So we wanted to compare by transcriptomics what's the impact of UCMS with the impact in major depression with some patients. So we get amygdala and prefrontal and cingulate cortex samples from human patients post-mortem that have depression at the time of the death, and we compare transcriptomic. And interestingly, we observe at this moment like a strong correlation in amygdala, very strong in amygdala, and also significant but less in the cingulate cortex. And it's what you see here, so like a molecular signature of stress and depression in this case. And interestingly, FLX is fluoxetine. When you give fluoxetine, not to the patient in this case, they have depression at the time of the death and they are dead, but in the mouse, you stop the correlation. So it looked like fluoxetine, antidepressant treatment, stop the correlation with a depression disorder in the patient. We also find in these studies that we have opposing neuroglial molecular phenotype induced by UCMS, and that can be reversed sometimes by antidepressant. And in this case was in mice, so we also have the dentae gyrus, so the hippocampal part. So we have a decrease, for example, in the dentate gyrus of neuronal marker phenotype after UCMS. Also, in the hippocampus, what we find is this, the UCMS, the number of genes that was affected by UCMS. You have this number of genes, and in purple is the gene that was reversed by antidepressant treatment. So half of the genes that was affected by UCMS was reversed by antidepressant treatment. And you have here the antidepressant treatment. We can see that fluoxetine, in this case, do much more than reversing UCMS, because you have all these genes that was affected only by treatment. So it's not like a mirror, it's not mirroring the negative effect of stress. Antidepressants do much more than just reversing stress, but it helps at least to reverse the stress effect. And we have, voilà, this is the pathway, but we see that very important for neuronal activity and plasticity. We have different genes and survival as well. Okay, so for the first criteria, it was okay. So then we wanted to have the second criteria, which is face validity, which is the ability to the model to reproduce a syndrome reminiscent of depressive disorder, so the depressive symptoms. So what are the depressive symptoms? So in human, according to DSM-5, so you have two important symptoms. The cardinal symptom is dysphoria and anhedonia. So anhedonia is the loss of pleasure, of interest for something you like to do normally in normal conditions. So you lost this interest. This can be measured in mice, we will see. And also you have some more somatic marker, like change in weight or appetite. It can be more, can be less in patients, sleep disturbances, psychomotor agitation or retardation. You have like fatigue, loss of energy or efficiency that decrease for the efficiency for doing like routine tasks. So you lose this energy and this efficiency to do routine tasks. You are slow, so you cannot do that. And you are more like psychological and cognitive symptoms, maybe more difficult to test a model in mice, but like suicidality. We don't have this in mice, it's hard to measure, of course. And guilt, worthlessness, concentration decrease can maybe be. So it's at least 5 of these 9 symptoms in human and present nearly every day for 2 weeks. But if you see, so with this criteria, it's a multitude of potential profile associated in addition with symptoms like anxiety, slight cognitive impairments. So we have one diagnosis for patient, depression, but actually an extraordinary heterogeneity in etiology and in clinical manifestation. Actually, we make the calculation. If we make all the combination, one patient that have the night symptoms, but with an increase of, I don't know, insomnia and not hypersomnia, we have 1,400 profile different potentially in depression, if we met all the combination. So, and we put everybody in the same disorder. So probably etiology, mechanism behind can be very different. So we wanted really to have a model that can reproduce like a spectrum of symptoms in the syndrome of the model, because we were thinking that maybe neurogenesis involved in one dimension more than the other, or one subtype than the other. So we wanted a model that is able to have the spectrum of symptoms at this moment. So UCMS seems to do like a phenotype, and we can test each this dimension with other model too, but like anhedonia, we use more the cookie test, which we will see a video later, but the mouse in 4 sessions should take a chocolate cookie, or the sucrose preference that you probably know, but I will not present after, but actually in the model it was not working really well for us at least. It worked well during the stress, but when you stop stress, you have normal consumption, so we never use it really a lot. We can measure body weight change, and actually we find sometimes, not always, another variability of the model, but a decrease of the body weight with the time passing over the weeks. But when you see weeks of UCMS, it starts to be different from stress to non-stress. So you have here the non-stress, and dotted line is the stress mice, to have significancy after 5 weeks of UCMS. And sometimes, not always, it can be reversed by antidepressant. So we have this also, some symptoms that I class in loss of energy or low efficiency of routine tasks, maybe. It's really speculation, but we can imagine something like that. Apathy, because we were able to measure the code state. So we see a degradation of the code state of the animal over the week. It depends on the strain, depending on the strain, but actually with bulbocele, like the white mice like this, you can see it really well. And it's also direct consequence of the stress procedure, but also maybe they are low efficient, they start to less groom during the home cage, which can also probably induce something like that. We have the splash test, so which directly splash sucrose water on the animal, and normally you have to induce grooming, and we can see difference in grooming, the same apathic, and also the nesting test that was sometimes testing in the procedure. We have a force swim test to test, maybe you know that, so the animal is in the force swim test, and we see the immobility of the animal. So we test sometimes tail suspension test is the same, so you are the tail is suspended and you see the immobility. Normally more depression is equal more immobility. It's not always the case with us, but it's what we can do. We also can test more anxiety-like behavior, like NSF test. So novelty suppressing feeding test is one that works the best in the model, for us at least. You have a conflicting drive between the animals that want to go for the pellet, food pellet at the center of a new arena. So you want to avoid the center, but also you have a drive for this pellet, food pellet. So we see the latency to go to reach the pellet. And so stress, we find this increase the latency, so make more time to get for this. You probably know there is open field, for you can do, we did also, like dark box, elevated plus maze, to test the anxiety-like dimension of the model. We also social interaction, we wanted to go into that. We use resident intruder tests that sometimes also work, and three-chamber tests for preference for an animal. So resident intruder, the test, you put an intruder in the cage of the stress or the control animal, and you look the antagonistic behavior between them, between the residents that should attack or not the intruder. What we find sometimes is that the stress increases the aggressive drive to the intruder. And that can be reversed sometimes by fluoxetine. Okay, we also make other sleep pattern can be used also, activity pattern during diurnal and nocturnal phase, and cognitive function. So also like Maurice Water maze, so we did, I'll present also later, but we have sometimes some also effect on but not always, and sometimes it takes different form depending on the strain. We also have some flexibility inhibition tests, so this can affect some executive function. And we particularly found like a deficit in inhibition task when you have to inhibit something you learn, to learn a new knowledge. But in this case, chronic stress, we find sometimes also that it can impact this inhibition in executive function, to have a new learning after one, and novel object recognition, we also. So you see, we have a large spectrum of symptoms we can test after, and sometimes we get some that's more replicable and other less, but we test over the year all these dimensions. So, and indeed, this UCMS can induce symptoms reminiscent of depressive symptoms. It's what so why we select this, like anhedonia, so we see with the cookie test, for example, body weight change, psychomotor, more the apathy, on va dire, the less efficiency with the code state and the splash test, change in anxiety, so for the emotional reactivity that change, and some cognitive impairment, and altered sleep. So the last validity is the predictive validity, and the objective is to see if antidepressants have the same efficiency in the model. What was also good in this model compared to maybe some biocell like force swim test, one shot of fluoxetine, for example, doesn't have effect in this model. You need to treat classical monoaminergic antidepressant for 2 to 3 weeks to have the first behavioral effect. So it reproduces a little the time frame we can see in the therapeutic response of antidepressant in patients. So that's why we selected that, because it checked many of the lists and the items we wanted to have as a model to see if hippocampal neurogenesis is involved, if it is the case, if it's more involved in some dimension or some subtype of depression. So we start then all these studies. So the impact of neurogenesis. So the first thing we test, it was if decrease of neurogenesis can cause the etiopathogenesis of depression, or at least increase the vulnerability to stress to induce some depressive symptoms. So we're still using the X-ray irradiation to inhibit neurogenesis. And so you have here, you can see in the subgranular zone, so of the hippocampus, after irradiation, and this is maintained for we tested 4 months, 6 months after, and we still have this inhibition for long term, and you have almost no neurogenesis compared to non-irradiated mice. And this is the other neurogenic area, the subventricular zone. It's focal, so we can see that the neurogenesis of the same animal is not affected in the subventricular zone. So the advantage of this method, it's that we are specific of hippocampus. After, it induces inflammation at the time of irradiation, so it's also a drawback. But compared to transgenic approach, the transgenic approach, you inhibit the two regions at the same time. So you cannot, it's a compromise between the two strategies. Here you are specific of hippocampus, just it was induced by a strong inflammation at the beginning. Okay, so voilà, it's the first study. So if you can see what we found in the paper, so irradiation worked really well, and here you have the number of newborn neurons, so that 4 weeks old, so they start to integrate the circuitry and to be active in the dentate gyrus of the hippocampus. And we can see from white to black here that UCMS decreased neurogenesis significantly. Not as much as irradiation, of course, but you have a significant decrease. So our procedure decreased neurogenesis. So then we wanted also, so it's just one, we also tested in anxiety for splash test or for cause state. It works, and even the weight, but I will show the result mainly to go faster on the anhedonia-like profile. So we use for the cookie test, for example, in these studies. So here you have an example. Voilà. So you have the chocolate cookie on the right. The cookie is here. So you see the mouse. It's accelerated. So voilà, she smells the cookie and she will go for the cookie in a few times. Voilà, up, she bites. So we have the latency, and we can also look how many times she bites the cookie. So we repeat 4 sessions, and you have the result here. So in white is the control animals, and in gray it's the stress animal, the UCMS animal. So we can see that over the session, so you have an increase of the cookie consumption in the control animal, but not in the stress animal. So it was the first point. So now we wanted to see if inhibiting neurogenesis will reduce the consumption or make them even reduce even more after stress. So you have here the result with the stress in dotted line. But we can see there is absolutely no difference with the non-irradiated mice. So actually there is no effect of irradiation for the control, the non-stress mice and the stress mice. Even the stress mice are now more susceptible to reduce the consumption. After we have here in this test maybe a floor effect, but it's the same in other tests. So actually, inhibition of neurogenesis neither induce a depression-like state, nor make mice more vulnerable to UCMS. Okay, so what we see, it's, voilà, here. Actually, it's not involved in triggering depressive-like phenotype from control, and also it makes not the stress mice worse. So it looks like it's not very so critical for the etiopathogenesis for triggering depression symptoms. But now we test the impact if it is at least important for antidepressant effect. So here now you have the results. So antidepressant like fluoxetine increase neurogenesis, so reverse the decrease of neurogenesis. Actually, non-irradiated mice have no effect in the irradiated mice. And now what we observe in the cookie test, and it's the same in other dimensions, some, not all, but you can see that fluoxetine reverse and increase the cookie consumption, even it's only stress mice with fluoxetine here. So we see that fluoxetine restore a normal consumption of the cookie, even in stress mice. But we observe this in non-irradiated mice, but not in irradiated mice. So it seems that without neurogenesis, antidepressant cannot induce the behavioral effect of antidepressant. So inhibition of neurogenesis prevents the antidepressant reversal of UCMS alteration. We found this also in the NSF test and in body weight change, if I remember, and the code state. So it seems that even if it is not triggering depression, it's not symmetrical at all, at least it's process that is recruited by antidepressant to contribute to the behavioral effect, or some behavioral effect of antidepressant. That was also confirmed by other groups, but sometimes with just some partial, not all the behavioral dimensions are affected by neurogenesis, but some, like maybe anhedonia, maybe some seem affected by neurogenesis. So, voilà. So if we make a summary of these first studies, was neurogenesis may not be pivotal for the etiopathogenesis associated with chronic stress, as we see. It did not induce depression-like states who don't potentiate the vulnerability to stress, but is required for the recovery effect in some tests, some dimension at least, of antidepressant. We also recapitulated with inhibition of BDNF. It's another line that we did, but it was very similar with antidepressant, but not to trigger stress phenotype. So these findings support the idea that neurogenesis has a crucial role for antidepressant effect while not causally involved in the etiopathogenesis. So then we were interested in the functional role, why this can be important in terms of some physiological process or neural activity that is relevant for depression-like phenotype. So we test directly that. So the first aspect was we wanted to look stress systems and how hippocampus can regulate stress system. First, so we look HP axis and particularly the corticosterone release in animals. So here you have like a circadian change of our mice. So in white is still the control animals. In gray you have the stress animal, UCMS, and in red the stress plus fluoxetine. So what we observe actually is not change in baseline, but you have like an advancing phase. So you have a change maybe in the sleep or the activity pattern. You have an increase of corticosterone in stress mice that come too early. At the end of the light phase, but normally it's more concurrent to the dark phase. So we have like an advancing phase that is partially reversed by antidepressant, not fully, but partially. And we have even an opposing pattern later during the dark phase. So it's really like the change of the shift of the circadian rhythm in our model. And it's bulbous mice. Ways is bulbous mice. Okay, and can be partially reversed. We also test the dexamethasone suppression test. So maybe you are not familiar with this, but dexamethasone is an agonist of glucocorticoid receptor. So normally when you inject the dexamethasone, you will have a negative feedback. You trigger the negative feedback, and normally some hours later, you have a decrease of corticosterone. So it looks the integrity of the HPA axis negative feedback. So normally you have a kind of suppression of the corticosterone induced by dexamethasone some hours later. So what we find in our mice, it's like a 50% suppression after dexamethasone in control animals, and you have a decrease after stress, significant, and that was reversed by antidepressant. So we wanted to test directly how hippocampus and probably neurogenesis can be involved in this aspect, because you have many glucocorticoid and mineralocorticoid receptors in the hippocampus, and it is known also to regulate, more or less, to have a regulatory control on HP axis in some aspect for hippocampus. So we test directly first the reactivity of hippocampus to dexamethasone when the mice are exploring a novel environment after UCMS. So we test with FOS, and we look particularly newborn neurons in the dentate gyrus or the mature neurons. So what we find, I'll go quickly here, but you have UCMS dampens a little the ability compared to control of the dentate gyrus to react to novelty and to dexamethasone. That is reversed by antidepressant. What was interesting in antidepressant, it's the part of the neurons that was activated by the condition, that was newborn neurons, is much more than the stress animal, but also the control animal. So it looked like during stress, so you have a less reactivity and less recruitment of neurons, and that when you have stress but antidepressant, we use in stress animal newborn neuron to recover this function that was maybe lost. So it looked like antidepressant recruit newborn neuron to maybe counteract what the stress have done of the reactivity and the recruitment of neurons. So it looked like it's maybe sensitive to this. So, and we also have to look the pathway where hippocampus can maybe inhibit and regulate the HP axis activities. You have no direct connection, but you have some nucleus that can serve as a relay to this effect. So we test some by injecting intrahippocampi the dexamethasone, and we look the activation in this nucleus. So what with FOS? So an immediate gene that track the active neurons. What we find without dexamethasone is very low activity. For example, here in the bed nucleus of stria terminalis. So this pathway here. And when we do dexamethasone in the hippocampus, we see an activation of this region in the control animals that is really reduced after stress. So after UCMS, you decrease the reactivity induced in the BST induced by hippocampus under dexamethasone. And this is reversed also by fluoxetine. So it seems that UCMS decreases the regulation driven by hippocampus towards stress systems like this nucleus and maybe the HP axis. So the last part of this project, we also we directly look at dexamethasone suppression test, but directly by injecting dexamethasone in the hippocampus. So and we have some irradiated mice again, so with no neurogenesis, and we look what happened after we implant cannula and we inject dexamethasone directly in the dentate gyrus. So what we find is the result are here. We have non-irradiated mice. We have irradiated mice here. So you can see in white in the control non-stress animals that you still, by injecting dexamethasone in the hippocampus, like a 50% suppression of corticosteroid some 4 hours after the injection. And actually, even with no neurogenesis, you have the same. So like a non-stress hippocampus is still able to do the job, to inhibit HP axis in normal condition. You don't need neurogenesis to do that. Under stress, you strongly prevent the ability of hippocampus in this configuration to inhibit HP axis activity after dexamethasone injection in the same way in non-irradiated and irradiated mice, so with or without neurogenesis. But again, with fluoxetine, you can reverse that in non-irradiated mice, but not in irradiated mice. So you cannot restore completely the ability of hippocampus to regulate, to control the HP axis activity when you don't have neurogenesis under stress. So it looks again that neurogenesis is recruited by antidepressant only under stress condition and UCMS to restore the hippocampal control and stress response. So what we find here to schematize, to have a schema of this. So what we find is that UCMS affects plasticity and neurogenesis in hippocampus, and even we have other plastic things in the change in the corticolimbic network in general. And this also strongly inhibits the ability of hippocampus to control the HP axis activity, which can result in lower negative feedback to this axis and excess or at least shift of the phase in the glucocorticoid activity under stress. And antidepressant, by first recruiting neurogenesis and maybe also other neuroplastic event, will be able to restore a little of this function, to regulate a little and restore this negative feedback, reduce stress system overactivity, and start to restore the normal functioning of corticolimbic network and maybe open the path to a remission. So neurogenesis will help antidepressant to promote the restoration of the hippocampal control and maybe of the corticolimbic network and stress system that opens the path toward a remission, probably in this model. So that was all this kind of work. And then we wanted to know, okay, we show that maybe neurogenesis may be necessary in some behavioral aspect and maybe for the regulation of the stress system, but is this sufficient? So now, in the last year, we're also testing now the augmentation strategy. If we just increase neurogenesis, is sufficient to induce antidepressant effect, which is maybe necessary, at least for some aspect, but it will be sufficient or not. So it was we did. We use more recently the IBAX mice. We are not anymore bulbous, but it's a mixed BLAC6 and bulbous and maybe 129 mice. So in this model, we remove, under recombinase and dependent of the tamoxifen, we remove the BAX gene of the progenitor cells. So in this case, we force the survival of the newborn neurons, because normally during neurogenesis, during maturation, you have a important part that is that die, that will enter into apoptosis. So while with this model, we can force the survival of during maturation of the newborn neurons and increase mechanically the number of newborn neurons and the neurogenesis of the animals. So it was we have here. So with this, we wanted to test, if by increasing only neurogenesis without antidepressant, we are able to find the same pattern we observe with antidepressant and of the different symptoms we have here. So we use, again, UCMS for weeks and different tests there. We find also that our procedure increases neurogenesis more strongly in stress animal. And we find, for example, in the cookie test, you have in dotted line, this is the latency. Well, this is the latency. So you see a decrease of the latency to go for the cookie in the cookie test. In full line is the non-stress animal, and in dotted line is the stress animal. So you can see that with more neurogenesis, you can decrease the latency that we don't observe without, with the normal neurogenesis. So increasing neurogenesis help in this case to improve in the cookie test. So you have like a antidepressant effect. But actually, the only one we find, because in all the other symptoms, even when you have stress effects, they did not reverse in the NSF, in the forced swim test. So we don't find many things actually. Even in the in HP axis, we just find, and it was opposing, so again, non-replicability. We have in this strain, and not with dexamethasone, but like 90 minutes, between 45 minutes, 90 minutes after acute stress, we have less corticosterone in the stress animal, the opposing we have with the bulbous mice. So, but at least we have an effect that is reversed by increasing neurogenesis, but it's the only thing we find. So actually, the augmentation strategy, we have no change in innate anxiety and hardly reverse depression-like effect and state. So just increasing neurogenesis is absolutely not sufficient to induce a strong antidepressant effect. So it's antidepressant effect is much more than just, it's a one lever. They can use it as one tool to help, but it's not sufficient at all. Okay. So then what we wanted to test after that is to see if UCMS, antidepressant, or manipulation of neurogenesis was able, but we saw that it can affect the biology of hippocampus, to reduce neurogenesis, to reduce other aspects, and have an impact on behavioral level directly. Then I was interested to know what happened in between. What is the impact on neurogenesis on computational treatment of the processing of the information in the hippocampus that can explain the behavior and the control of stress system? Because we know the cellular effect, we know the function, but what happened really? What do neurogenesis and UCMS to the computational property of hippocampus and how we code the ensemble activity and dynamic in the hippocampus? So for this, we directly look activation, we look spatial and temporal encoding in the hippocampus directly during task. So for example, we use electrophysiology part, so we can record animals during some tasks. This is an NSF test, and we can, and also some open field exploration. And we find, for example, here a plast cell, so a neuron in the hippocampus that is active only in this part. We did also similar thing with calcium imaging. So here you have, we can record the activity of neuron by calcium activity. So here you have a window, a field of view of the hippocampus during a task, and you can see some neuron flashing, so directly recording from neuron during a task. After, we can make a footprint of the field of view and get the trace of each neurons during the task and observe, for example, this neuron was only active in this area. And you can also generate the heat map of the activity of the neuron. So we can really create what neuron was active during a task and how it codes the space, for example, during this task. So this is really preliminary too, but what we find is like particular antidepressant under stress can improve the encoding of novel environment when you look how they will encode new environment and enable a faster place field formation and cognitive map stabilization during this moment. And also, antidepressant improves the encoding of slight contextual changes. So you have, with this, promote the rate remapping, so it's really specific, but we change the rate depending on the context, and it reduces interferences in the code we are able to extract from this experiment between overlapping experiences. So what we believe here, it's finally the enhanced capacity to encode novelty we find with this recording, help to disambiguate some contextual changes and respond to stress stimuli may result in getting the downstream brain region, like HP axis, to more accurate information under antidepressant when you have stress. So it improves the way to encode the information and to have more accurate information for the downstream brain areas that are involved in stress system, and that can then improve behavioral adaptability and stress coping, for example, and reduce overgeneralization. So voilà, this is the full picture we get for UCMS, but we see at the end with the augmentation strategy that we start to get very different behavioral patterns made by UCMS. So all the first part was at the beginning of my PhD and the postdoc, and then the augmentation strategy when I came back to France 8 years later. And we have very different effects of UCMS, and we see that the strains that serve as a transgenic model was not the same we use at the beginning, and we have different patterns. So we start to see a strain effect. And actually, one aspect, so I will present now, we also tested the strain comparison. We make a while ago also compare 7 strains in one procedure in the UCMS. So it's what we have here. So we compare in different strains. So this one is the first studies here, but we have another with the Swiss mice. We have 7 strains, and we look physical activity, physical state, locomotor activity, anxiety behavior, and HP axis. And with AG mice, bulbous-C, C3H, black-6, CBA, DBA, and FVB. So this in code state, you have usually an impact on the code state by UCMS. But you can see that the effect size is completely different from a strain to another. Black-6, it's really hard with the black-4 to see actually the impact of the code state. Maybe there is no, maybe they are more resilient, or maybe the color also that make, because in white, like the bulbous-C here, you can see really well the difference between UCMS. So first we are very different in the term of effect size of the stress, and then you have in gray here the effect of antidepressant, it's imipramine in these studies, and you see that in some strain you have a reversal effect, bulbous-C, but not in almost in all the other nodes except for DBA. You have a reversal effect. So the antidepressant doesn't work in this model in 5 out of 7 strains. First aspect. So, voilà, we have just 2 that works. NSF test, that's the same. Finally, it works. We have a stress effect in AJ, bulbous-C, and black-6, and not in the other strain. And for the 3, you have a reversal of the antidepressant effect. But again, it's only 3 strains out of 7 strains. We also look fecal, so baseline cort is not the best, but we did that at this moment. The basal corticosterone of in half day, which was we collected feces for half day. And also we have many strains where you have no effect, but you see sometimes, at the time at least we take the feces, we have the opposing pattern between bulbous-C and black-6. Again, completely opposing effect. And some reverse, and also opposing between DBA and FVB, decrease in DBA and increase in FVB. And some reverse, in red you have the 2 strains when you reverse by antidepressant, but the 2 other was not reverse. So we have a very multifaceted picture depending completely on the strain. Here you have a table that summarizes what I just showed. Also in another studies, so I go faster here. In another studies, we also look in the same strain, the Morris-Water maze. So the impact of chronic UCMS on Morris-Water maze. We have 4 strains where you have an effect of stress, but if you look the effect, it's completely different from a strain to another. For example, bulbous-C have a deficit in the first day of learning, and at the end, not at the middle. While AG or CBA is the opposing. You have the same level at the beginning, the same at the end, but you just have a learning curve that take more time to reach the optimal performance. And also, black-6 also have a decrease at the beginning, but after they have really good performance at the end. So again, we have a completely different picture that is strain specific. So it's a multifaceted strain specific effect. maybe we have some MDD, some say major depression disorder subtypes that originate from different genetic makeup. We should maybe go more into it. We have other divergent lines that maybe represent more this subtype of depression. And we were also, it looked like each readout, behavioral, cohort, etc., are independent because we have no correlation actually with one pattern in corticosterone and the effect of NSF, for example. We don't find the same, well, it's not the same strains that have the same impact on HP axis will have the same result in the NSF. So we don't find a correlation in these studies at least between one dimension and the other. Well, after in these studies, the range of alteration was investigated was quite limited, so we don't continue this aspect, but it will be maybe worse. So, so last part of the talk. So I think for this workshop particularly, but I collected the data before, but I did really for the talk today. We were thinking a while ago in the lab we collected all the data, all the procedure we made from my PhD, and even when I was not in the lab of Catherine Belzung lab, we collect the data, and when I came back after, for 20 years actually of using UCMS in the lab. So I found 38 procedures, the data of this. And that's why that back to the lab in 2016, while I was there for 8 years, we've been seeing a lot of variation, decreased effect size in experiment involving UCMS procedure compared to what we have at the really beginning, which was already variable, but not at this point. So voilà, we have many studies done with that. So we said, well, it's time for a full assessment, like an internal, a lab meta UCMS. So it was we did, and recently. So why we did that as well? But more than one experimental facility during these 20 years. The lab changed 5 sites of animal facility. There is 2 different during my PhD, and at the very end experiment also a new one. And then when I come back, we have also 2 other sites, and we have a new site again. But the new one, I did not make UCMS since that. But so we have different sites, and for each site, we have very different pattern, well, not very different, but different pattern of result. We have more than one UCMS procedure. It changed a lot, actually, during the years. This one, the very first one, it was 10 weeks. After, for example, in 2008, I used a 5 weeks one. After, for the molecular psychiatry paper, I used a 8 weeks one. So it changed a lot, and the same for the stressor. The combination of stressor, the identity of stressor, changed a lot with the years. So this one with Ducote, but voilà, this was we did with IPEC. And we have another one. This one was maybe the more severe with the combination of stressors, so we can go more in detail after, but voilà, we have very different procedure. So I wanted to see that actually. Voilà, fiber location, multiple change in housing, gooseberry regulation is an important point because something we can do during my PhD, now it's not possible, particularly with control animal. If I have time, I will talk about it, but the way we care, the control animal, changed completely from what we can do at the beginning of my PhD. So this can change, ethical standard change. Also, I will come back to this, but during my PhD, we have a real unpredictable stress. It means I was PhD student, I come at midnight, at 2 o'clock during night to do a stressor, and now we are attached to the working hours. We cannot go during night, ask people to go as we were doing when we were a student at this time. So the unpredictability is not the same because the amplitude where a stress can come, we cannot do the same as we were doing at this moment. It's completely changed. So all this working regulation changed as well. So we have a large number of different experimenters who are involved, and minor, also major change in the experimental UCMS procedure. As I told you at the beginning, it was the intensity of individual stressor was the same all over the weeks, while in the in 2011 paper, we have a gradation, an escalation of the level of stressor and the combination. Because after you have it's mild stressor, and in the name it's unpredictable chronic mild stress. And that's why when you have 6-8 weeks, actually it's very minimal stressor, and the mice habituate, they are familiarized, it's not so, it's even enrichment no, maybe not, but maybe not. But it's it's something that happens, and they know the stress. So that's why we also have some procedure with an escalation of the stressors, the duration of restaurant stress, the duration of without sawdust, and we combine sometimes without sawdust with social stress. So it's all over the 8 weeks. So the escalation was also, we will see, is really good, but it's more labor intensive. But in varying conditions for control animals, this is super important too. It changed a lot. And also at the beginning, at this time, we were exclusively on male mice. Now we work systematically with females also. But social stressors are absolutely not the same impact in this UCMS on female and on male. The social stressor is not so stressful for female, while in male, with the male aggressivity, it can induce some stress that is completely different. So the objective of this, so it's really new, but identifying the protocol characteristic and readout associated with higher effect size and better replicability. I'll try to find some. And so I determined this quickly. It's really new, fresh, better. Determining effect size of the UCMS effect, and then also I will present antidepressant in UCMS. And after, including some moderator or subgroup among the possible sources of variability. So the sources I identified, but we can help me to improve this, because it's just really preliminary. We use mouse strains, sex, facility, experimenters, and within procedure, make a level of severity, which can depend also of stressor escalation. It's what I just explained. We have a escalation of the stressor over the week. It's really soft at the beginning and become more and more to avoid the habituation. The variety of the stressor, the time window of the stressor delivery. As I said, now we cannot have the 24 hours in predictability. It's just 8 hours of the working day. The duration of the procedure, the administration route of the drugs will change a lot. And after, the control conditions that change, how you handle control mice. If you have a cage, change mode of the cage is changed also with the ethical regulation act. Now we have to change more frequently than before, and actually it's really stressful for the mice. But voilà. Group or isolated control, enrichment, and catch size. So with this, I have, as I told you, 38 procedures and 2,209 mice in my meta-analysis. And this is the result I obtained for these 20 years. So you have, in the first part of the UCMS, so the ages, so the combined effect size for this criteria, for the UCMS effect, and here the antidepressant effect in the UCMS mice. So you can see with time we have a strong combined effect of the code state score, body weight gain, so you have a decrease of the body weight gain due by stress, it's negative. The NSF test was one that worked in this, but not splash test, was why at the beginning was one of the ones that was working really well, but actually over the years it's completely almost zero. The light dark box, FST, actimeter, the cookie test is there, but there are only 5 studies with, so it's not a lot, so we'll see. And nesting, resident intruder, no significant effect, but just 3 studies. BRDU for 4 weeks old newborn neurons, it's significant, and plasma baseline cohort is not significant. I haven't sufficient data for the negative feedback. And we can see the antidepressant effect. We have 3 where you reverse, at least partially, for cause state, body weight, and NSF. And we have an effect of antidepressant in splash tests. But there was no, so it looked independent of stress because we don't have a stress effect in this. So after, we have some effect in cause state, for example. This is what we observe for the cause state. So we have almost all studies, you have an effect. But as I'll show you, most of the studies here are with bulb C. If you do this with black cyst, for example, the black cyst probably is this one, you don't have a really significant effect with this on the cause state. And particularly what I believe, when you look, I go back just here, the effect size is much bigger for UCMS than the reversal effect. Because actually, I think really the cause state is a direct consequence of the application of each acute stress on the cause state. When you stop, it starts to be better. So really the variance explained by the stress and what antidepressant can restore work a little with bulbous, but it's not the full change of cause state. So I'm not sure it's a really good indicator actually, and particularly if you are not in bulbous mice. So this is a cause state, voilà, with antidepressant effect. So you can see that the confidence interval frequently cross the red line. So actually there is not so much studies where you have this antidepressant effect. When you have the combined, you see significant, but actually maybe half of the studies did not detect a significant antidepressant with cause state 2. Now it's weight gain. Again, so you remember weight gain, you have a UCMS effect in the meta-analysis, but again, there is not so much studies where your confidence interval do not cross the red line. So it's not, many of the individual studies is unable to detect a change in the weight gain, and we will see in the moderators, but it's really dependent of the severity. Actually, body weight is really dependent on the severity of the procedure. And we cannot make the same severity now as before. So it's so this is the antidepressant effect in weight gain. So it can reverse, but you can see it's all. So it's significant when you combine, but actually there is very few studies how you reverse that. And when first you have the first aspect, but you have maybe these two data, this as well. The first one here. You reverse, you have an effect of antidepressant body weight gain, but not when you look all the 38 procedures, you don't find it so frequently as well. So again, a big variability in the result actually from each individual studies. This is an NSF test. So we have a very weird studies here because the only ones that go to the opposing effect. I don't remember what studies it is, but those are, so we have some, maybe half or a little more detect a UCMS effect in the NSF test. So maybe the best behavioral test for UCMS is the NSF, the novelty suppressing feeding test is the one that have the best effect here. And it can be reversed, not always, but for antidepressant effect. So I go a little faster now because I have to finish. But we can discuss later on the detail if some are interested into that. So I just go to the conclusion of this. Voilà. So the source of variability. So we can see with this, so I will not point, but I put the moderators, as moderators, strain, sex, in the meta-analysis. So I just summarize what I obtained. So mouse strain, of course, is a full, is an important factor that explain variability in the result. And as we are suspected from the beginning, bulbousy is the strain that respond better to the UCMS, even if it is not huge. And sometimes we have no effect with bulbousy, particularly in the recent procedures that are softer, that are milder. But it's the ones that have, when you have effect, bulbousy have a better effect side of the effect. Sex also is important, as I explained. Female are maybe less sensitive to social stressor particularly, so it makes the range of stressors that are really efficient maybe less important, so it can impact, for this. The facility, in our case, when I look, the 5 facility we use, we have huge difference and explain a lot also the variability. So how to explain? I'm not sure it's really the variability, the facility itself, because actually we change and Catherine changed the facility over the years. At the same time, they change the procedure. So it's explained by the site, the environmental condition of the facility, or the procedures that change over the years. So you have a confounding factor here. We cannot be sure it's really the facility, but when I put it like that, it has a huge effect. Experimenter, no, this is a good point. So for the procedure, severity have a strong effect, particularly on body weight, I told, but so effect size, the severity increase the effect size when it works in NSF, for example, and the body weight change. It's important in a way because when I started with UCMS, people were telling it's not the severity of the stressor, but the unpredictability, which is a real factor that influence the result. But actually, no. When I look at the combination of stressors, the duration, severity also play a big role in the result. So stressor escalation of the variety of stressor also, as I explained, because maybe it limits the familiarization, have effect, and when you have a good stressor escalation, the combination, it increases effect size, and it's one that improves replicability. When you have a good stressor escalation, actually the results are more stable in this case. Time window of stress delivery, so I'd say it's 24 hours or just a working day. But it's not as much as the previous one, but it has this effect too. But it goes a little with stressor escalation too. Duration, not so much actually, just for the familiar, because maybe when it's short, they don't familiarize, so you have some effect. When it's longer, with no stressor escalation, longer is not better, but when you have stressor escalation, longer may be better. So that's why I think it's counterbalance depending what you do. Administration route have also some effect actually. If you put the antidepressant of vehicle in the water or you inject intraperitoneal or peros, can change a lot, and particularly it changes the control actually. So voilà, because you will handle the control daily, while it's in the bottle, no. And we will see right now that for the control, by look at the control condition, so usually it was always in a separated room in our case, handling act a lot. Actually, when notably the first procedure we used, we wanted to disturb the less possible the control, so they were much less handled than the stress animals. But when came the behavioral test at the end of the procedure, the control are not used to be handled. So they have very acute stress, very strong, and it completely blurs the chronic stress effect. So handling the control was super important, and it's also maybe what administration routes, when you have to make IP injection every day to the control too, makes a better difference, actually, that's when it's in the bottle. But if you prefer to use in the bottle, you have to, each day, go to your control and link them, and maybe this will avoid the handling effect, the difference between control and stress animals. So low handling reduces the effect size in behavioral tests because you have an important acute stress in your control animals. Group and isolated, also we are counterintuitive because particularly we remember in the neurogenesis I have to implant cannula. So my control for all the experiments is that we are isolated. I don't group my control for this experiment. And some other. And actually, I have better replicability and even a good effect size in isolated control. It was counterintuitive because we were always thinking that isolated the mice was a stressor, so we want to avoid isolated control. But actually, I have more replicability and stability in my results when my control was also isolated. There was big cage, some visual contact with the other, but not physical contact with the other. Group induced higher variability. And what I said also, it's because before, I think it was a speculation, but I think it's really have a role too. When in the PhD, when the control enter into the experiment, we stop because now we change the cage in the animal facility 2 times a week when you have 4 or 5 mice. So completely you change the cage, you completely change the sawdust. And you can see sometimes, particularly Balbucy, but also other, that when you have the new sawdust, new cage, the dominant have to reestablish the social hierarchy. So you have some aggressivity that increase at each time you change the cage. So at the beginning of my PhD, when we have the control, we change really few the cage, maybe once a week and not 2 times a week. And also we always let some spot. You know, when you do like this, you have some spot with urine. Also you have some house, some house in some smart cage inside the cage, and we keep the same. Like this, you have the urine odor maybe of the dominant, and we have much less aggressivity in the cage. Now we cannot do that for hygiene, for hygienic condition, and maybe it's also explained because if you change the baseline stress of your control, of course the delta between the stress effect will be less, and you blur the power of your result. So, have a good control. It's maybe what impacts more UCMS because control change a lot. So it was just explained with the cage change mode. So this has an impact on enrichment where also in cat size can play. Okay. So in conclusion, we can see that strain, procedure stability and severity, and control condition matter a lot. So we can go more into detail after for the discussion. But when control conditions are already stressful, the effect size is reduced of your model, and UCMS lost all the contrast it can have if you have this aspect. And this is super important because if UCMS is small and unstable, then by investigating the treatment, the reversal effect of a small delta, it's super hard. And why me, I'm really interested in the mechanism. So if my UCMS have a small effect, and I want to see an antidepressant effect, and now we are interested to block the antidepressant effect. But if you cannot first see the antidepressant effect, you can even less block the antidepressant effect. You need a big delta to another big delta, and you lost everything. So you cannot investigate circuit if you don't have a very strong effect size and replicables. So it was we have here, investigating mechanism, blocking a reversal become untestable in this condition. Okay, so I was a little long, I'm sorry. Voilà. So I want to thank, of course, Catherine Belzon, my supervisor, my PhD, and now director of the institute, Arnaud Tanti, my colleague. We work together now in Tours, and all the persons that participate to these studies and the collaborators. So thank you very much. You have some picture of my city that we took at the dinner, so you are really welcome to visit. It's really nice. Thank you.
So we have time for 3 questions.
Alex, a very great talk, and I will follow a little bit on it in neurogenesis. So if you look at antidepressants, yes, so normally what we do, they alter neurotransmitter uptake, yes, né? But on the other side, né, the question is, if you could treat depression and increase neurogenesis just by using an anti-inflammatory agent.
Oui. Mm-hmm.
Yes? Yeah?
We try anti-inflammatory, not me, but in the lab we try.
Yes, so this would...
no.
But on the other side, nah? So neurogenesis is also a neuroinflammatory process. It is not strictly anti-inflammatory, yes?
Right.
So if you, that was a question, so maybe, so I know depression is neuroinflammation, yes?
Right.
But is neuroinflammation so bad? So may not be neuroinflammation be involved into the repair effect as a compensatory effect by the brain.
No, but it's possible. We have a line of research of our colleague Vincent Camus, and I was involved. We have a review paper on this. It's really nice. You can read it. And it did not work well in the model, but we have some P2X7 knockout mice we use in the UCMS model, and we have contrasted results. Sometimes we have an effect on decrease on neurogenesis, otherwise not so. And we try with anti-inflammatory agent, and it's contrasted result. But we were thinking exactly to test this question, but we have contrasting results, so I have no define very well response to this, but it's an axis that we wanted to develop.
So one more addition. So it is not only astrocytes and microglia which will be activated. Nah? Is it also, if you look in the peripheral blood, yes?
Where?
In the peripheral blood, do you have an effect of hematopoiesis, for example, yes? Mm-hmm. So where is basically coming this activation?
I don't know. I don't know where it comes, but we have IBA1 increase. We have in the brain, we see this marker change clearly in the hippocampus. It's the work of Romain. Thomas Gosselin too, was a PhD in the lab. So we test the activation and the ratio between non-activated and activated glia in the brain. But the origin, we don't know.
Thank you very much.
Another question?
Thank you, Professor. Sorry. So actually, I'm curious. 20 years of UCMS, that's quite impressive. Congratulations. That's a serious relationship.
It's not only me. I have a hiatus of 8 years, but the lab, there is people that continuing it. So,
Yes, and what is your secrets to keep students motivated? Because it's a tough protocol, you know.
Because it's a very labor-intensive procedure. At the end of the PhD, they were really what you say, angry against me to involve them in the UCMS, but I use it not a lot now. We use it since I'm back for the augmentation strategy. It was the work of Uda Eliwa, which is, and Luca colleague. So they were really work intensive and with IBAX mice. But we use it much less. As I explained, I'm very interested in the mechanisms, actually. It's what drives my passion to this. And I feel that at the moment we cannot use the same procedure as before for this regulation. And I really want a model that will have big effect size. So here I cannot rely on UCMS now. So I use it much less now. So the current students I have tell me, thank you.
No problem.
But you know, actually, what we use with the calcium imaging require a lot of training to explore. It's almost more labor intensive than UCMS.
Oh, wow. Lovely. So my question is, have you looked at, because you brought at some point in your data the PVN, yes. Have you looked at anything related to early life stress?
The PVN?
Yes.
The activation of PVN,
exactly,
for example, with
PVN,
FOS
no.
after, you mean?
Because some studies, people shows, for example, as a shift to start with early life stress and then switch for UCMS. So you have done something.
We haven't looked, but the one when we were looking for, I show the relay nucleus, like BST. At this moment we look, but we don't find a difference. We have an effect of dexamethasone, but even not so much. I don't remember now, but I have to look the data. But we look at this moment, the first change after acute stress, and actually we don't. It's an effector, so maybe the regulatory system that will change in terms of FOS. Also, you know, FOS, the same for hippocampus, but FOS tell you the recruitment of neurons. So you just, you know, it's a yes or no, but sometimes it's just the quality of the code, the identity of the cells that are involved that can change, and not the summation of the activation. It's why I say to my students sometimes, when you look for human fMRI, you can see some regions that are involved because you have a different activity in this region. Exactly. But the negative result doesn't mean this region is not involved, because you may not have a net effect of the total of activation, but the quality of the coding and the encoding of the activity may be changed. For example, the work I presented yesterday, or the one with I don't present the detail, but with the remapping, with the recording, I show also here. When I say you have a faster cognitive math, that is the formation of the cognitive math become faster with antidepressant, and you discriminate better to context. For example, when you change the context and you have an effect of stress of antidepressant, actually, if I look FOS, I don't think difference. But what I can see is the code in the hippocampus. The identity of the cells that are active will not be the same, will be more different after antidepressant. You have less overlap of the cells that are recruited. But the global activation, if I just look the number of FOS, I see the hippocampus is not involved. No, that's completely wrong. It's super important. But just when a mouse will less differentiate the environment, it will have an overlapping in the code, an interferences, because it's the same identity of cell, or some dimension, but the same identity that is active in the two conditions. But when the animal can really well discriminate, it will completely recruit a new different cells. So voilà, it's maybe PVN also has some, I don't know, but maybe you can just have a shift of the activation, but not a global change of the total amount of activation.
Yeah, absolutely. I was quite impressed about that, because you mentioned the effect after the antidepressant in dental gyros. Right. Because in some studies that I follow up, there's a real huge actually oscillation in some cortical limbic circuit, particularly in those that pass through the early stress. So, and then what is the age of the animal that he uses for this? Yes, for the antidepressant fluoxetine. Just curious.
Right. And first, also, this PVN is really work with acute stress.
Okay. Okay, yeah, because
and then the age is usually we start they have 2 or 3 months. at the beginning of the
Ah,
irradiation
uh-huh.
or the beginning of the stress procedure.
Doesn't
So
make
they
sense.
go until it's 8 weeks sometimes, so 2 months more, and then the test, so they go to 5, at the end of the experiment, they are 5, 6 months.
Oh, right. Okay.
Yeah,
Okay.
to 2 to 5, 6 months.
Yeah, exactly. Okay. Thank you very much.
Thank you very much. Next, fascinating data, and I love it. Is this last part published yet?
The last part, no.
Oh, but
You see, the
it
meta-analysis
will, I
is
hope.
internal. I
It
don't-
will, no? But it's very, very interesting. So I have a question regarding this last part. Fascinating. You have all this strain, this mice strain difference. So my question is, well, the question I also have is about the translational aspect.
Right.
So what would be the ideal mouse in terms of translational aspect? And then I wonder, all these strains, they are, well, very different from humans in the way that they are very genetically
very
similar and so on. So maybe the ideal mouse to do translational work would be the mouse we catch on the
Right.
streets, because that's something very important to consider when you
Right.
and your data reinforce that. So I would like to just listen to your thoughts about that. Thank you again for the
Right, well, that
wonderful
would be
time.
very interesting. But it's also, this make me think that, something I forget to say, but that's right, that we can see, at least with the 7-strain comparison, it's also give indication for translation. It was having the same etiological factor, which is chronic stress. And we can see that chronic stress, the same etiological factor, induce very different pattern of alteration from a strain to another. So it looked like, well, maybe also in human, it's what we can observe sometimes, the etiological factor will maybe not track certainly one patient to one specific subtype of depression, of pattern of symptom, because at least here we see that the same stressor, chronic stress, because it was the same procedure, moreover, send in different paths the different strains. So the genetic also have an important way how the clinical manifestation will appear even with the same etiological factor. Okay, but thank you very much. Okay.
So now we have a coffee break, and after coffee break we will have Professor Henning, and I don't know how to pronounce your last name, you have to teach me. Ulrich, yeah, good. So I was right. Ulrich, right? Yeah. So please enjoy the coffee break. The coffee break is sponsored by Tor Labs. If you have a moment, please go to them. They have like amazing and tiny microscope there, so you can see what they can do and what we can see inside the brain of the mice, similar to what Professor Alex just showed you with the calcium imaging. Thank you so much. Live? So, back to our talks this afternoon. So our next speaker is Professor Henny Ulrich. He is a full professor and head of the Laboratory of Neuroscience in the Department of Biochemistry at the Institute of Chemistry of the University of São Paulo, here in Brazil. He studied biology at the University of Hamburg in Kiel, in Germany, and complete his master thesis in biochemical parasitology, followed by a PhD in biochemistry and neuroscience at the University of Hamburg. He later carried out postdoctoral training in the Center of Molecular Neurobiology, also in the University of Hamburg, and at the Cornell University in the US. If you, by any chance, have been interested in preinergic signaling, probably you read not one but several papers from Professor Henning. He is a specialist in the preinergic system and canine signaling, and he served as the president of the Brazilian Society of Preinergic Signaling. His research focuses on neurogenesis and neuronal repair mechanisms, both in vivo and in vitro, with emphasis on canine and preinergic signaling. He has established new conceptual frameworks in the field of preinergic research, and he's also a pioneer in using aptamers and for identification of stem cells and tumor cells, and he has like a huge contribution in drug screening, calcium signaling, flow and imaging cytometry, and he really supports young investigators. So thank you so much, Professor Henny, for accepting our invitation. It's an honor to have you here.
Professor Aline, thank you very much for this very, very nice introduction. And now I will tell you a little bit about preinergic signaling, yes? So, I will not discuss any stress protocols, but I will show you that preinergic signaling in the beginning has all to do with stress. So, and I also would like to mention, yes, so with INCT, this is a National Institute of Science and Technology, and we are supporting research groups who are interested in working in preinergic signaling all over Brazil with a lot of international collaborators. Thank you very much, Alina. So now, the idea of my research during the last 25 years, when they're coming to Brazil, is actually to understand how stem cells can transform themselves into neurons and form a neuronal network. And also, so how they contribute to a certain type of a neuron and how these neurons can be protected against injury. It means stress. And looking at neurodegeneration. So stem cells was in the beginning, and neurotransmitter receptors. In the beginning, I was also working on other types of receptors, including nicotinic acetylcholine receptors. So now, let's start out with preinergic receptors. So ATP is very well characterized as energy molecules produced mostly in mitochondria. But when ATP is released into the extracellular space by different mechanisms, maybe exocytosis, which may be channels, or which may be just that the cell dies and releases ATP. Inside the cell, so the ATP is in the millimolar concentration. Under healthy condition, extracellular ATP is at nanomolar, maximum micromolar concentration. And this has also to do, so But ATP released here by a cell, for example, by an astrocyte, it acts on polynergic receptors. P2X receptors are ligand-gated ion channels, which are activated by ATP. Yeah? And they cause currents by influx of sodium and calcium and the efflux of potassium. And then ATP and other nucleotides, like UDP, act on P2Y receptors, which are G-protein coupled receptors, which have a slower activation and slower desensitization phase. So what you see here, that ATP is immediately degraded into ADP, AMP, and adenosine. And adenosine receptors are also G-protein coupled receptors. So the idea is now here that there's always a balance between ATP, which is excitatory and pro-inflammatory, and adenosine, which would be more immune suppressive and more inhibitory in many contexts. So now, with polynergic receptors, so just to introduce a little bit, the P1 receptors are the adenosine receptors, where you have 4 subtypes. I'm also working on them, but due to time limits, I will not discuss adenosine receptors. I will focus on P2 receptors, which are activated by ATP. And what you see here, that you have P2X receptors, so which are trimeric ion channels formed by subunits, by 3 homomeric or heteromeric subunits, while the P2Y receptors have in mammalians 8 types of receptors. And just to advance a little bit, I will talk here only about 2 receptors, the P2X7 receptor, so which I define as a bad receptor, which will cause neurodegeneration, cause stress, while the P2Y receptor will alleviate stress. So it's a neuroprotecting receptor and a receptor inducing neuroregeneration. So now, what you see here, the idea, why I can talk maybe today here at a symposium on stress. What you see here, what happens if ATP is released at high concentrations, yes? You see this here. It will activate receptors, yes? And it will also, activate what you see, P2X receptors at high concentrations. And there is a member of the P2X receptor, which is the P2X7 receptor. And this receptor has characteristics different from the other ones. So it can form a pore. It's not clear how the pore is being formed. But with the formation of a pore, so it may let through molecules until 700 Daltons or maybe even bigger. So it can directly release cytokines. And it can let flow into the cell a huge concentration of calcium, uncontrolled, and killing the cell. And what you see here, that all this, I will go into more detail about that. Now that the P2X7 family, yes, that you will have underlying signal transduction, and what they form, that you have formation of reactive oxygen species. And most interestingly, so that a major function of the P2X7 receptor is the induction of the inflammasome. So the P2X7 receptor induces the inflammasome, and the inflammasome causes the release of inflammatory cytokines and caspase. So it means it is a cell death pathway. And also, what you see here, there are new results. I will not talk about this, but you find also the P2X7 receptor in the mitochondria. So now, going to the next slide. The concept is why stress with ATP at high concentration, so if cells die, so inside the cell is 5 millimolar of ATP. It will release this ATP outside. So it will take the extracellular ATP concentration to the millimolar range, maybe. And this is called making ATP a DAM, a danger-associated molecular pattern molecule. And it says, there's danger. And it will activate the innate immune system. And this leaves here, basically, in the release of inflammatory cytokines. So it mostly, so the P2X7 receptor, which I will talk about, so it's a doubt if neurons express these receptors or not. But for sure, it's very highly expressed by any type of immune cell and astrocytes and microglia cells. So what happens finally, that is that the P2X7 receptor, by a huge activation, turns the microglia cells into reactive microglia and into pro-inflammatory responses. So now, the idea is that I will give you one example here using in vivo model, and then I will go to stem cell models, which are also used for modeling neurodegenerative diseases. So as Aline said, we are doing both of it. So first of all, I will talk about the cytotoxic effect of P2X7 receptors in an animal model of Parkinson's disease. So now, Parkinson's disease, as well as other age-related diseases, they come from an activation of the immune system. So you don't have only acute inflammation, so inflammation becomes chronic. And what it says here, so that when we are young, we have a homeotic adaptation. So there may be inflammation, but it will go away. And the organism is fine. But if you go for a consistent inflammation, which in case of neurodegenerative diseases may initiate in the gut, so if gut inflammation or wherever, and at a certain time, so this transmits into the brain. Initially, what is expected, that you will have a dysfunction of a brain-blood barrier, so that you have an invasion of peripheral immune cells, which will then induce microglia cells and make astrocytes reactive. What you see here, and what you have in this, basically in this process, that you have a release of pro-inflammatory cytokines, and finally, what you have is a redox dyshomeostasis, and, for example, a death of dopaminergic neurons, which causes Parkinson's disease. So, Parkinson's disease, everybody knows well, I will not spend a lot of time, but what is very important is that it does not start in the brain. It may start in the gastrointestinal issues, and finally, from these non-motor skill symptoms, you will become the motor skill symptoms like, for example, rigidity and postural instability and all the tremors and the dysfunction of the motoric system. And this is caused, what you see here, by the death of dopaminergic neurons, the motor symptoms, and also the missing dopamine release and stimulation. So, and you see that with non-motor systems, they will take into motor systems tremor, rigidity, and imbalance, basically based on the on an imbalance of the basal ganglion. I will not go into that, but the purinergic system, the purinergic signaling, is not only causing, involved in this neuroinflammation process, it is also directly involved into the imbalance of basal ganglion D1 and D2 receptors. So now, what you see here about the proinergic receptors, the P2X7 receptor. What is a very important point, what you see, it is mostly a cell death receptor, but always you have in this P2X receptor, you have different functions. It may, in some stages, even stimulate proliferation, but at low ATP concentrations. But if the ATP concentration go up, we will kill the cells. And it has clear functions in embryonic and adult neural progenitor cells, because it maintains the pool and it prevents the formation of brain tumors. Okay? But you see also, on embryonic neurons, it induces apoptosis, and also in microglial cells. And what you see here, and on the right side, exactly this idea, if there is an ongoing lesion, then, so with dying neurons, or they release a lot of ATP into the outside. And this ATP basically will activate P2X7 receptors on astrocytes and on microglial cells, and increase the lesion, always increase the lesion. So in view of that, it may be therapeutically indicated to inhibit this receptor. So now, we see here, but the thing is not so easy, because I told you the receptor is bad, but on the other side, if the receptor may basically block proliferation to protect the system. For example, if there is a transplantation of stem cells into the brain, so it may, this receptor may inhibit uncontrolled and damaging proliferation. This is a major factor also in the healthy brain. On the other side, the P2X7 receptor, which I will not show the original data of my lab, but they're very clear. So if we go for neurogenesis, in the beginning we have high P2X7 receptor activity because it may even induce proliferation of stem cells. But when we come progenitor cells, then the activity or expression of a P2X7 receptor needs to go down, that cells become neurons. If the P2X7 receptor stays high in activity and expression, cells will differentiate to astrocytes. Okay? So the idea is that it regulates neurogenesis and stem cell integration. And what we see here, because this I wanted to discuss with you originally, so it was very clear that we have 2 points of adult neurogenesis. This is in the dentate gyrus, in the hippocampus, and in the subventricular zone. What do we know now today? That this cell may migrate somehow. And you find also adult neurogenic zones in the substantia nigra, in the cortex, in the striatum, the substantia nigra. You are aware the dopaminergic neurons die in Parkinson's disease. And the process of, maybe I need to go one back, of neurogenesis is basically a neuroinflammatory event, because neuroinflammation regulates synaptic pruning. You need to have apoptosis. You need to redefine. So I mean that neurogenesis works together with neuroinflammation. So finally, I will show you some data. I will show you the idea of this work that we did. First of all, if you have dopaminergic death in Parkinson's disease, So we propose to inhibit the P2X7 receptor in the animal, and whereby we will inhibit the cytotoxic form of the receptor expressed by astrocytes and microglia cells. But also, neural progenitor cells may have a receptor. And if they have a receptor, the P2X7 receptor, there will be no neurogenesis. The cells would differentiate into glial cells. And now, the idea could be different strategies to try to neural repair in Parkinson's disease. One possibility would be clearly to inject stem cells into the brain. But in this case, you have many times that you form necrosis. The cells will not integrate into the system. And because of that, a concept could be to stimulate endogenous neurogenesis. Yes? And this has been done with GDNF, has been shown that it could work in principle in neurodegenerative diseases like Parkinson's disease. But what I'm proposing here is that we inhibit the P2X7 receptor and look for neuroregeneration. And as I showed you, so we have defined other neurogenic zones, that was shown 10 years ago by Jonas Friesen, that there are basically neurogenesis in the striatum, in the adult human brain. That means that this proposal could be valid. Okay. So now, the idea is now how we do with experiments. So we use a rat model, okay, and then we use 6-hydroxydopamine, inject this basically into the nigrostriatal pathway to induce dopaminergic death. And what happens is actually that 6-hydroxydopamine acts on the complex 1 of the respiratory chain and induces ROS, induces stress, and with the stress leads to the death of dopaminergic neurons. And okay, so we do this as a unilateral injection. Only in one hemisphere we inject the toxin. And this means that when we challenge with animals with apomorphine after a time, after 1 week, we can see if they have motor symptoms. And with motor symptoms is if we knock out one hemisphere of a dopaminergic system and the other hemisphere is okay, then with animals they start to rotate. We have a rotating behavior which we can use as a behavioral test to assess with dopaminergic degradation. And what we see also, now here we use for staining for dopaminergic neurons in the striatum and the substantia nigra by staining for tyrosine hydroxylase. So this is, and what we see here, that the models which are valid. Now we have on the injected hemisphere, we have almost a total loss of dopaminergic neurons. Yeah? And we can see this, that we have basically, but we have after 1 week, we have a huge loss, and it is the same after several weeks. It's the same thing. So that we could work with a 1 week period after injection of 6-hydroxydopamine. Okay. So now, what we are looking for, also to justify our project, is to look at different P2X receptors which are expressed, and then looking in the presence or absence of injection of 6-hydroxydopamine. And what we see here, with the receptor, which is largely changed in its expression, is the P2X7 receptor. First, there's a huge lesion. That means that receptor expression goes down. But then, with scar formation, the receptor expression goes up again. So it means that this receptor is regulated and could have a function in dopaminergic cell death. And now, what I said to you before, what the idea is, that the neurogenesis is guided or it's prevented by neuroinflammation. So if we inhibit this neuroinflammatory receptor, we could have recovery of dopaminergic neurons. And we use for an P2X7 receptor antagonist a compound which has been approved by the FDA as a food, giving color to food. So this is brilliant blue G, and you see we have a structure of brilliant blue G, and this is a mostly selective inhibitor of the P2X7 receptor. So now, how we do the assays. So on day 0, we have a unilateral hemisphere lesions by 6-hydroxydopamine. Then we wait for 1 week, and when we do, we challenge the animals with apomorphine and do the rotational test. And then we treat with brilliant blue G, and after another week, 7 days, we are going again for a rotational test. And if what we expect, that the animal rotates less than in the beginning. And finally, we go for immunohistochemistry staining for inflammatory markers and for tyrosine hydroxylase. Okay, what you see here, maybe I need to click on the slide. Is it working? No. Yeah, you see it. It is working. Yeah? And now, go to the next slide. I go to the next slide. You see, this is the idea of this rotational test for Parkinson's disease. And what we see, go one slide. What we have, so the day, basically, 7 is the day 0 for BBG injection, because 7 days after 6-hydroxydopamine injection. And we see that the rotational behavior goes down after 1 week and after 2 weeks at a concentration of 50 milligrams per kilogram. And now, if we increase the doses, when you see a milligram per kilogram, what we see that at 75 milligram per kilogram, so the rotational behavior is gone. That means suggesting that we had neuroregeneration. And what we also see in immunohistochemistry assays, where we see always to the control of the unleashed area is here that you have tyrosine hydroxylase staining. So if you inject 6-hydroxydopamine at a low concentration, in the absence of BBG, there is no regeneration. But now, if you increase the concentration of BBG, then you see that you have a regeneration and staining for dopaminergic neurons by tyrosine, stained by tyrosine hydroxylase. Okay, you see this in the striatum, and this is, you see the same in the substantia nigra. Okay, that means just to quantify this by what we do by quantitative tissue cytometry, but at a concentration of 75 milligram per kilogram, you have a total regeneration of dopaminergic staining. And this is in line that the animals do not rotate at all after 1 week of treatment with this concentration. Okay. And this is also in line with the reduction of the neuroinflammation, which you see here we did an IBA1 staining of activated microglial cells. And if you treat the animals with BBG, then with inflammatory staining is almost abolished. So it means that P2X7 receptor inhibition favors neurogenerative and neuroprotective processes in a rat model of Parkinson's disease, okay? So it means it is very good to inhibit the P2X7 receptor. Now I come to the second part of my talk, where I will show you a little bit about neurogenesis. It is all in vitro, that extracellular ATP drives neural differentiation and regulates sulfate through differential purinergic receptor activation. And what we basically, the justification for this assay is that on one side it's very good to have animal models, but on the other side it's also very good to work with very simplified conditions where you can look at a specific condition of activating or inhibiting a purinergic receptor. Yes? I mean, what we use here is we use multi or pluripotent stem cells which, upon stimulation, differentiate into neurons. And looking at that, the idea in these experiments is always that we have different sources of stem cells. So what I will show you here today is mostly with embryonic stem cells or with iPS cells. But we are also using primary cultures of neurospheres, which we then plate as neural stem cells, and when we differentiate and look at the conditions which happens upon pharmacological or genetic modulation. And with this works very well. It shows here, for example, a neural progenitor cell stage. So we induce for neural differentiation. And what you see here, but here are the undifferentiated cells, and you have the cells are stained by DAPI, but you have a migration of the cells like you have in cortical development. And this is, you see in neurogenesis, you have a network of neurons, and you have a network of astrocytes. And finally, you have a neural network which is similar to what happens during cortex differentiation. Now, the idea is, in this first work, that you used a mouse embryonic stem cell line, a feeder-free mouse embryonic stem cell line. And also here, what you see, if we induce this cell with retinoic acid, when they form basically networks of neurons, as you see here, stained by a neuronal marker TUJ1. And why these cells are good, I show you here. Because with embryonic stem cells, they go through all phenotypic stages of neurodevelopment. So in the beginning, they are basically positive for OCT4. So this is for undifferentiated for embryonic stem cells. And then they go into the neuroectodermal differentiation by stage-specific embryonic antigen 1. And finally, they express nestin, which is a marker of neural stem cells, yes, on day 4. And then on day 8, they become young neurons and young glial cells. And day 16, they are mature neurons, marked by MAP2 and mature astrocytes. So now, what basically has this to do with poietic signaling. And what I wanted to show you here, maybe we could just, it's not working, maybe we should click on it. Okay. The idea, one more. Sorry about that. Why doesn't it? Oh. Okay. So the idea is, this is a very much research question of mine. So is how basically poietic receptors induce differentiation. And what we know, that stem cells and undergoing to a differentiation to neurons, that we have a rhythmic calcium oscillation. And with rhythmic calcium oscillations, they come from very low concentrations of neurotransmitters secreted into the extracellular medium. So that we do not desensitize the receptor. So you have an oscillatory activity. And what I show you here is basically that in part, this oscillatory activity is due to poietic signaling. And if we look now at wave, with oscillatory activity, we can see that we have two types of activities. So one is a very quick, very rapid activating and inactivating amplitude. So this would be a spike activity, yes? And then you have lower activities, with lower transients, but they take more time, they are present, which is a wave activity. And it makes sense that we say that the spike activity is mostly due to P2X receptors, because they are neurotransmitter receptors, while the P2Y receptors would be more responsible for the wave activity. Okay? And basically, so it's always a little bit slow here because it takes some time for changing the slide. An ATP, a long time ago, we showed that ATP, this is by BRDU staining, yes, and increasing concentration of ATP, and using pharmacological context, we could first show that in undifferentiated cells, ATP induces proliferation. Yes? And basically, as the first step, so induce proliferation, and now it also induces differentiation. Yes? So to understand this here, so here's a staining for nestin and for new specific enolase. Yeah? And what we see, that ATP induces that, and any interference with intracellular calcium signaling. For example, by BAPTA, which complexes intracellular calcium, EGTA, which inhibits calcium influx, CPA, blocks, interferes with intracellular calcium release, and so on. And the general blocker of poietic receptor, PPADS, all of them block this differentiation activity. So now, based on many different types of works, we could come to the conclusion that the P2X7 receptor in undifferentiated cells, as it does also in cancer cells, may induce proliferation. And then, if cells become to differentiate, what goes up here is the P2Y2 receptor, which I mentioned in the beginning, while the P2X7 receptor goes down when we differentiate into neurons. Okay? And we showed this by RNA interference experiments with cell lines. So now, the idea, and this has been not only seen by us, but this is a paper by my group, that in healthy conditions, what happens, that we have an upregulation of a P2Y2 receptor over the P2X7 receptor. Yeah? So the P2Y2 receptor is the good guy. The P2X7 receptor is the bad guy. And then, in disease condition, we have a stronger activation of the P2X7 receptors, which inhibits neuroregeneration and leads to neurodegeneration. So, and the idea was here to show this in a context. We used the embryonic stem cell lines, and we implemented a protocol for differentiation into GABAergic neurons. And what we can see here, we use 2-SUTP, which is a selective agonist of the P2Y2 receptor, versus BCATP activates mostly P2X7 receptors. And what we could see, under this condition, we can increase the differentiation rate into GABAergic neurons. Yes, what you see here by basically by gene expression, you see this by flow cytometry, and we have by immunostaining and so on. Okay, it means the P2Y2 receptor favors GABAergic differentiation. And now the question is, to make the thing more interesting, what comes after the calcium signaling? There comes neural transcription of an expression of specific proteins which characterize GABAergic neurons, okay? On neurons in general. And what we use know here, that I call this mesh 1, ASCL1 is mesh 1, and neurogenin 1 and 2, they may act in antagonic functions. So my idea is, just we look at this one, but these two factors determine GABAergic differentiation. While has oligo ID and oligo 1, 2 are for glial differentiation, okay? And the idea is now that looking at this hypothesis, what could work, but we have two types of receptors. We have here the P2Y2 receptor, which induces calcium signaling from intracellular pools released from the endoplasmatic reticulum. And then we have the P2X7 receptor, which induces the calcium influx, but the idea is that the P2Y2 receptor activation basically will, through activation of chymodulin, kinase, etc., activate MESH1, while also the P2X7 receptor will show you will activate neurogenin 2, because the calcium signaling patterns are different. Both of them induce calcium transit. But it depends on the type. It's like a signature, which type of signal transduction will be activated. And the idea is, okay, but why again ATP? And what we see here in the left panel, that during differentiation of the stem cells, that we have high ATP concentrations, extracellular, relatively high. Now, you see this, now maybe 200 to 400 nanomolar in the extracellular space. And this is necessary before the cells are terminally differentiated, which happens on day 16. So there, the ATP is not anymore needed at risk to induce that. So its concentration decrease. And what you see here is basically, which are differentiated stem cells, embryonic bodies, and they have a very high calcium activity. This is it's not a no. And now, okay, that is fine. But we would like to go ahead with this. So what I propose here is a neural clock that calcium and gene expression triggering differentiation as a neural clock. And it is well known that MESH1 is oscillatory. So differentiation does not go on. It stops. But if mesh 1 transcription becomes sustained, differentiation goes ahead. Okay, and the idea is now the following: how we can do experiments. So this I did with Ryoji Kageyama from Japan. So basically, creating transgenic mice. And these transgenic mice have new mesh 1 or neurogenin 2 linked to a luciferase reporter. So, generating with transgenic animals and then isolating embryonic stem cells and differentiate them into neural progenitor cells. And this condition, we can do the following. Here shows a little bit this idea, how this can be done, that we image calcium in the second time, a minute time, and then we look at gene expression in the terms of days or hours. So we very fast calcium imaging, and then we have a mesh 1 luciferase bioluminescence or chemiluminescence imaging. And what you see on the top is the calcium, the oscillating calcium, and what you see on the down is that neural transcription factor expression is oscillatory, such as calcium oscillatory. And now we want to create a link. So in the following experiment, I think this presentation is very heavy, and that does not. Next slide, Aline. No. Huh? Next slide, yeah? Yeah. Yeah. It's okay? Next. So I'm sorry, but I give you a problem now here. Yeah, this one, no? Yeah. So now the idea is the following. What you can see here is we do calcium imaging, yes, and we record the spike activity at very low BC-ATP and 2S-UTP concentration. 2S-UTP activates the P2Y2 receptor, BC-ATP the P2X7 receptor. And what you see again, what I showed before, that any interference with calcium signaling, basically by tapsigagine, which inhibits intracellular calcium release, and it depletes the intracellular stores, or EGTA, will basically, you don't have any mesh 1 expression. But interestingly, UTP expression accelerates the sustained expression of mesh 1 compared to the control. So it means it's faster expressed, so it will be induce neurogenesis. While on the other side, BC-ATP activating P2X7 receptor does not activate mesh 1 expression as we expected. And now, in the next slide, we did the same with neurogenin 2. And what you see here is the opposite what you saw with mesh 1. 2S-UTP does not activate neurogenin 2 expression. On the other side, BC-ATP induces neurogenin 2 expression. And what we say here, that we have a balance. And actually, what you see here, with this GABAergic differentiation, basically is sustained under these conditions of 2S-UTP stimulation. And again, what we can see here, I showed you this before, but 2S-UTP actually induces GABAergic differentiation, as you can see here, by GID 65. And most importantly, looking at mesh 1 over neurogenin 2 ratio, what we can see here, with 2 as UTP increases the mesh 1 over neurogenin 2 expression, while BCATP activating the P2X7 receptor does the opposite. Okay? So, okay, this is great. But it also opens therapeutic applications, because there are diseases in which GABAergic neurons die. So what we can do for increasing the number of GABAergic neurons? And so the idea is that we look now at Huntington disease in stem cell models. And basically, what I will do, this is the last part of my talk, the idea of Huntington disease is that we have a mutation in the Huntington protein, yes, and what you have an increased number of polyglutamine residues. And if they are basically less than 35, you're okay. If you have 36 to 39, you may have the disease or not. And you have more than 40 of these polyglutamine residues as a mutation, then for sure you will have Huntington disease. And the more of these residues you have, the earlier you will get it. And with Huntington disease, has basically, so it's like uncoordinated movements, but it has also effects like depression, aggressiveness, psychosis, and cognitive loss. So it has psychiatric, it has motoric function, dysfunctions, and it has psychiatric and memory dysfunction. And finally, it will lead to death. So now, if we look at this mutated Huntington gene, again coming back to stress, what you see here, that anyway, that you have a polyglutamine region that you see here. And this is necessary for interacting of this Huntington protein during development with transcription, regulating transcription. Yes? But if you have a too long expanded polyglutamine region, what happens? That these proteins cannot be anymore removed from the nucleus into the cytoplasma. So it basically stays there, and it creates a stress. So this is one thing on transcription regulation. And then it is known that it interferes with intracellular calcium signaling, again, causing stress. So you see, what you see here, the normal Huntington does not do anything, does not interfere really. But if you have a mutant Huntington protein, it interacts with inositol-3 phosphate receptors, and it also interacts with mitochondrial function, and it interacts with calcium channels, okay? So you may think that you have an overflow of the cell by calcium causing stress, yes, and causing cell death. And most interestingly is that also the purinergic synthesis and signaling is disturbed. I don't go here very much into detail because these things are not understood. But you see, at some points, so you have basically you have increases and you have decreases in enzymes and also in the production of purinergic compounds. So I won't make any point here. But how can we study this? So we use here two types of approaches. The first one, we used iPS cells for Huntington patients. and compared them to control cells, iPS cells from healthy people. And then we came back also to our mouse embryonic stem cell model. And what we did is basically we knocked out the endogenous Huntington protein. And then we overexpressed the wild type protein with 23 repetition. So this should be healthy. It should be okay. And then we alternatively we put in a disease-causing protein with 74 polyglutamine residues, okay, using the CRISPR-Cas9 technology. So now, very interesting is that you need the Huntington protein. If you just knock it out, what we also did, then we go from we go until the basically the gastrulation state, and then it's over. This embryos will die. And what we basically, what you say, that the knockout of the Huntington protein is lethal before complete gastrulation. And the idea is what we increase in these conditions of a knockout NF-kappa B positive cells, and this NF-kappa B during development is basically preventing apoptosis. And the embryo needs to have apoptosis, as I said to you in the beginning of my talk. And if you block apoptosis, you cannot eliminate any cell, and you have no space that you can occupy by new cells. Yes? And also, Huntington, the apoptosis plays a major role in shaping and sculpting the embryo after the gastrulation starts. So we are writing a manuscript about that. Now, sorry, Alina, it's a little bit slower. I think we are okay here, yeah? So, what is also very interesting here in this context, so what we use, what we look first, basically on what happens to basically to if we express with this it's again, if we express the mutant protein during development, when we see first of all that basically, so this is this condition, that we have an overexpression of this protein because it cannot be really eliminated. So we have a very high expression of this Q74 compared to Q23, okay? So this causes already stress. And then we have a second point that we have under these conditions. So the Huntington protein is involved in the expression of brachyury and with brachyury positive cells. Brachyury is basically coding for mesodermal development. Under these conditions, with 74 polyglutamines, you get a shift from neuroectodermal differentiation to mesodermal differentiation. Okay, so I think this is a point that very interesting. And now going to our famous spike calcium oscillations. So now here we use the iPS cells. And what we see here, that in neuroprogenitor cells, so from Huntington patients, that you have a huge decrease in spike activity. And also, so you see this in the frequency, and you see this also here in peak height. It means that Huntington mutation interferes with intracellular calcium signaling. And now, to understand the next slide here, yes? So we use on day 8 of differentiation, and basically we are differentiating NPCs from Huntington disease patients. And now, if we look at this point here, what we use here is control against tapsigagine. And what you see in the tapsigagine, the pre-incubation with tapsigagine inhibits the calcium ATPase in the endoplasmatic reticulum. And it will basically deplete intracellular calcium stores, and it will flow the calcium into the cytoplasm and basically increase hugely the intracellular calcium concentration. And what we can see here, under conditions that in Huntington disease patients, mostly that we have an increase by tapsigagine. So we have here 0 days and control. Then you have together with tapsigagine, you see a huge increase here. So we have a control and the tapsigagine condition. That means that you have an increase in propylidian iodide positive cells indicating cell death. Okay, so the cells become more sensitive to high calcium concentration. Okay, and they die. So it means there are 2 points. One is that you have a higher increase in calcium, and the second is that the cells also become more sensitive. Yes? And here is done the same thing with annexin 5, marking cell death. So we have a control and a Huntington disease control, and then you have here basically in the presence of tapsigagine, where in control cells it goes even, you don't have any positive effect on cell death, while in Huntington disease you have a huge increase. Okay? And now, going on the right side, where we see that we challenge with ATP, yes, in neural progenitor cells, and we see that we have a higher response in the Huntington disease patients. Okay, so it means altogether you have more calcium in the cells, and the cells are more sensitive to high calcium concentrations. Okay, now basically coming back to the mouse embryonic stem cells, okay, and coming back to our mutations that we put in. And what we are looking here at purinergic signaling, at ATP and other agonists, so which I did not show you here, but at 100 micromolar and at 1 micromolar, so there is really, there is no big difference between control and Huntington disease. There is one difference, yes. And what you see here, basically, principally in the stage of neurons, if you activate the cells with 2S-UTP. And what we have done many times, and we could show that in embryonic stem cells, which were transfected with the disease-causing protein, the P2Y2 receptor activity is reduced. It's not the P2Y2 receptor expression, because we did always experiment, but there is an interference, specifically with the P2Y2 receptors, and with P2Y2 receptor we showed before, is responsible for GABAergic differentiation. Okay? So, I mean, the conclusions, that we have spike-like oscillations that were absent in neuroprogenitor cells from Huntington disease patients. And the Huntington disease model points at interference of Huntington mutation with P2 receptor-induced intracellular calcium responses, what I showed you. And also with SALDAF sensitivity. And P2Y receptors responses are induced following overexpression of a disease-causing protein. So this was a very nice work that we published in Molecular Psychiatry. And finally, just to put everything together, what I showed you during my lecture is that P2X7 receptor inhibition favors neuroregenerative and neuroprotective responses. It is neuroregeneration because we induce SALDAF. Yes? And then we put the inhibitor. Okay? And the cells come back. So there, again, you have probably neurogenesis and you have new dopaminergic neurons. Okay? If we use another receptor inhibitor of a P2Y6 receptor, which I did not show you here, there is no effect if we inhibit the receptor after induction of SALDAF. Where the inhibitor is effective if we co-apply it with 6-hydroxydopamine and prevent SALDAF. Yes? So this is very clear. It's not really a neuroprotection, it's a neuroregeneration. And the second part that I showed you, that P2Y2 and P2X7 receptor will regulate the cellular fate of neural differentiation through calcium and neural transcription factor oscillations. And the P2Y2 receptor drives GABAergic differentiation through activation of MESH1 and has therapeutic applications possible in Huntington disease. And purinergic receptor activity modulation as therapeutic strategy for neurodegenerative diseases. So actually, there are several clinical trials underway. Yes? Some of them are positive and others not. And as I say to you, the P2X7 receptor, if you just plug out the receptor response, so many times in neurodegenerative diseases, in human trials, you did not have a very good response. And this is due to the fact that the P2X7 receptor is expressed by every immune cell. And if you just remove the activity of this receptor, yes, then you have a number of side effects. Yes? So here, inhibiting purinergic signaling, you need always to have in mind that these purinergic receptors are expressed by almost every cell, and not only in the brain. So they regulate metabolism, they regulate proliferation, and immune responses, and so on. Okay? And finally, I would like just to thank my group, so who has done a lot. We work with Huntington's disease that we published in Molecular Psychiatry was Talita Glaser, while she, I think I don't have her here, Agatha Oliveira, she a former member of my lab. She did most of the experiments in Parkinson's disease. And I would like to, of course, to mention my international collaborators in this project. So with Ryoji Kageyama from the Riken Center of Brain Disease in Japan. With him I was working on neural transcription factor oscillations. Yang Deteng is a specialist on iPS cells. Yong Tang, I'm working together with him in China on neurodegenerative diseases. He created a big science center on pruinergic signaling, so I have grants with him together. And my local collaborators are Claudiana Lameo and Luis Brito from the University of Sao Paulo. So thank you very much.
So we have time for a couple of questions. I will start. So I was really curious about like everything. First, congratulations. It's like a lot of data. Everything is so well connected. So it's really an inspiration. But you said that at first people believed that only immune cells were like able to express adenosine receptors like P2X receptors, but then almost every cell can produce. But I was curious about like the role of these receptors in neurostem cells and neurodevelopment, because in the case of humans, we have like at least 5 weeks, there's no microglial cells in the brain. So do you think that maybe these receptors can be important so then neurostem cells can act, they can defend maybe the central nervous system and act like immune cells as well?
Yes, so the idea is, so first of all, and maybe it was misunderstood, so pruinergic signaling was only accepted in the 90s, yes, because before everybody, ATP, this is an artifact put on cells, yes, and this was done by the work of Jeffrey Bernstock, yes, and he showed first the effect of this receptor in neurotransmission of P2X receptors. And this only became credible after the cloning of all these receptors. So these receptors needed to be cloned, yes? No? And now you ask about?
I was asking about if like in the neurostem cells.
Oh yeah, I published another, I was also working on the side with bradykinin receptors, and always also were involved in inflammation, yes? And we were using neurospheres from mouse or from rat, and we did transcriptoma, Alzheimer's disease versus control. And what we found, that with neurospheres, yes, they have already an inflammatory potential, yes, because they express many genes, yes, which are involved in inflammation. And now I was also reading a little bit more about that. That neural stem cells, they have some immunomodulatory functions. So they are able to secrete cytokines, yes, this is well established. And because of this, you can also call neurogenesis as a neuroinflammatory process, yes? Because in the beginning, I was not really understanding that, no? And I got a critic on one of my papers, no? And so I said maybe there are some microglial cells, no? on embryonic day 12.5, I don't know, during neural differentiation, yes? But what I see now, what is more probable, that when neurosphere by themselves have this immunomodulation, so, and they liberate pro-inflammatory cytokines, yes? Yeah?
Perfect. And anyone else? Questions? So that's the second one.
So I make everybody tired.
So how is cell stress can be translated into stress, like chronic stress? And do you think like how could we modulate like cellular stress to improve its stress response or prevent its stress response?
Okay, so cellular stress. Now, if I look at the purinergic system, yes, so when ATP, in principle, now if you would induce stress, yes, adenosine would reduce the cell stress, yes. What you could do is basically you could increase activities of with actonucleodases, which transform ATP into adenosine, which would be one point, and also reducing the cellular stress on the other side, because very clearly P2X7 receptor induces cellular stress to inhibit this receptor, P2X7 receptor. This is the point, because what is stress? It is different types of stress. But what I talk about here is oxidative stress. So that you increase oxidative stress, yes? And with this oxidative stress leads to damage, to cellular damage, yes? And also increases, by this again, reactive species. So this is obviously one point, and other types of cellular stress in this content. I see also that you increase the intracellular calcium concentration over on more than heavy values. So if a high calcium concentration, this is clearly cellular stress, because when the calcium goes into the mitochondria, and you will alter the membrane potential, and you will affect ATP production, yes, which leads to cellular stress. Yeah? So the idea is cytokines, there is basically calcium is a cellular stress, yes, and also through the P2X7 receptor a disbalance of ionic concentrations, because I did not go into, so let's in the sodium, but it flows out with potassium, yeah? So it disturbs the ion concentration between inside and outside of the cell, which are also defined as a cellular stress. Yes? In neurons, with cells may not even be able to respond accurately to stimulation for action potentials and so on.
Thank you very much. Anyone else? No? So thank you again. So I know everyone is exhausted, but we have another task here. So we would have like a round table, but because everyone, it's like, I can see you're exhausted, and I'm exhausted as well because of this morning. Yeah, no, it's because of this morning. And I think that the main goal of this workshop, it's going to be tomorrow, in the end of the day, when we are going to, this is not my idea, this is the Professor Deborah Sushek idea, that I think we are in the moment that we can create a network in Brazil, a Brazilian network of stress research. So in the outside, we have a QR code that you can scan, and then it goes direct you to a form. And you, if you want to join the network, Brazilian Network of Stress, you can put all your data there, and then we are going to collect your data to contact you after the workshop. But tomorrow, we need to discuss what we are going to do about the stress protocols we have. So, and in this brief moment, I just want you to try to think about all the difficulties you have in your daily lives dealing with your stress protocols. So in any moment, do you feel like you don't want to work with the stress anymore just because in the end of your protocol, you have too much variation in your data, or because the readout you're using, the behavioral test that you use in the end of your protocol, it's just not as Professor Alex showed earlier, that it's not demonstrating the effect of your stress. So just tell us, please, What do you think? You don't have to tell us today. We can go straight to Invicta. Invicta is a beer place here, so we can go there, we can drink beer, and while we drink beer and eat something, we can discuss that. You can bring us your ideas, you can share your struggles with your stress protocol. And then tomorrow, we need to stay here a little bit later so we can discuss these and see exactly where we go as Brazilian network of stress, right? Can we do that? So everyone knows where Invicta is? Yeah. So it's really close to the university. Even if you don't drink, please go there because it's a moment we can socialize and we can meet you and we can talk. I can really learn your names. So please go. It's really important for us. It's a network to go. Okay? So I'll see you tomorrow at 9. I promise tomorrow is not going to be as stressed as today. It's going to be easy. We are going to start with a yoga class with Professor Vanessa Soares from the Federal University of Rio Grande do Norte. So it's going to be quite different. Thanks so much for your patience today, and I hope you enjoy your first day of workshop.